Hi everyone, and thank you for joining in for the Synthetic Biology Club hosted by the Abdel-Mawgoud Synthetic Biology Lab. This monthly online event aims to promote discussions around synthetic biology and also to share its latest scientific advances. The format of the event includes a presentation of up to 40 minutes, followed by a period of question and discussion of up to 10 minutes. Today, our presenter who kindly accepted the invitation is Patrick Diep, a PhD student at the Biozone Center for Bioengineering and Applied Bioscience, and also founder of the Canadian Synthetic Biology Education Research Group. He'll be discussing the state of synthetic biology education from the perspective of iGEM students in 2019, with his presentation titled Solving Disciplinary Silos in Synthetic Biology in their Undergraduate Curriculum Development. Patrick Diep is a senior PhD candidate at the University of Toronto as Biozone Center for Bioengineering and Applied Bioscience with prior training in biochemistry and synthetic biology at the University of Waterloo. He studies how metalloproteins interact with metalloion species in complex solutions for the purpose of protein-based nickel recovery from mine effluents. He's also the co-founder of Lyrita, a startup that 3D prints reusable soils for hydroponic farming operations that has received $200,000 in pre-seed funds. And he's the program director and founder of the Canadian Synthetic Biology Education Research Group. So before we start, I would like to remind you to keep your microphones turned off in respect for the presenter and keep your questions for the end. And now I leave it to you, Patrick.
Great, thanks, Ben. I'm going to share my screen. Okay, turn my pointer on. So hello, everyone. I'm glad it's a smaller group, actually, because I'm hoping to get a lot of feedback and questions and critiques on some of the stuff I want to present today. Really, I hope that you can feel free to critique everything that I talk about and do. As a fellow grad student, I'm totally okay with that because my professors critique me all the time. So I'm used to it. I'm sure you will be used to it too. What I want to talk about today is not so much hard technical science, so much as it is educational research, thinking more about, as a student, what are the students' learning experiences, learning environments for synthetic biology in Canada, and what can we do to make it cooler, make it more fun, make it more interesting, prepare them for more, dynamic working environments. So the title of the presentation today is Dissolving Disciplinary Silos in SynBio, Undergraduate Curriculum Development. I'm presenting on behalf of my group at CSBERG. And I wanted to introduce myself a little bit more. Ben, thank you for that very comprehensive bio that I wrote. So I'll just fly through this. And what I want to highlight with my introduction is that through these different experiences I've had the pleasure of working on, I've gained different skills in different kinds of fields: biochemistry, aqueous chemistry, which has a lot of the math there with the mass action kinetics and modeling, with rapid prototyping, so microelectronics, CAD design, 3D printing. The startup world has deeply immersed me into this entrepreneurship field, the idea of managing people, sales, and lastly, CSBERG. This area of research that is less quantitative and more qualitative / mixed methods, a lot of project management, and of course, teaching. So the backdrop of this presentation is I have been able to try my hands at many different things. I'm not an expert in, I would say, metalloprotein analytical chemistry. I'm not an expert in 3D printing or mixed methods research. I'm just familiar with a whole bunch of them, and I know how to coordinate people to be able to work with me on these different things. And this will kind of come up again in CSBERGs in the later parts of this presentation. So the first thing I want to do is talk about problems. What I've been able to get practice on is working on different types of problems in different kind of fields. And the way I think about problems, there's three ways to look at it. The first way I wanted to explain problems was through this quadrant here, which I'll also draw a little bit on. Just make sure it's there. Go. So the first type of problems are the most simple types of problems, where you start somewhere, you have an end goal, and the path to get to that point is pretty obvious. So the steps to get to the end goal are obvious, and the path to get there is obvious too. You can imagine if I have an expression vector to express a protein, I would put it into BL21DE3 probably and express that particular protein. So that's one kind of problem. Another type of problem is where you have your starting point and your end point, and you have no clue how you're going to get there. For example, if you want to, engineer a non-model organism, you do not know how to engineer like a tardigrade or some specific, niche microbial species. So this is another kind of problem. Then you have this problem where you know where you are, you know where you want to be. I should say clearly that before, you don't know the steps, but you know the path. In this case, say you want to engineer a microorganism to produce biofuel, for example. There's all sorts of techniques you can deploy to get to that point. And you know that probably somewhere on that path, you will have to deploy some computational methods, a little bit of intuition, and your path will not be obvious. You'll go in this area, and hopefully at some point you end up in your final product. So the steps are, where you need to go approximately, which steps to get there. That you need to do computational methods, but the path is just not clear. In this case, the best example I give is metabolic engineering. And the last example I want to give for problems is the problem where you have, where you are, and you probably know where you want to be, but the path to get there and the steps to get there are kind of not obvious. And I don't have a very solid synthetic biology example for this. The best example I can give is, imagine you've been working in the lab 9 to 7 p.m. You come home, you're hungry, you're looking for dinner. You come home, and in my example, I have my girlfriend, and I don't even know that she's mad at me before I get home. I get home and she's mad at me, and I have no clue why she's mad at me. I don't know where to even start to figure out why she's mad at me. This is a real example. So I started searching for the house to see what I did wrong. And I go to the dishes and I find out that I didn't wash the dishes, right? These are like unknown unknowns where you don't even know what was wrong in the first place, and you don't know where to start looking for that problem, right? The best example I can probably think of for synthetic biology are the fluorinated compounds you find in plastic wrapping. There's some startups in the US like Alonia from Ginkgo that want to degrade PFAS compounds, these fluorinated compounds. The question is, where do you even start looking for enzymes that can remove fluorines from carbon atoms? And which microbes would you even look for? It's hard to kind of tell where to go. And so you're kind of just left confused. This kind of quadrant, it's the known unknown quadrant that you're probably hopefully familiar with. If not, this is what it looks like. Sometimes you know exactly what the problem is and you can get there. And then there's these two versions where you have missing pieces of information. And then there are problems where you just have no idea like where to even start, or even if a problem exists, right? This dimension of problems, we'll call it knowledge. And so this first dimension is kind of getting across the point that problems require people to engage with different forms of knowledge. You can think back to synthetic biology that there's different forms of knowledge that needs to be engaged oftentimes to solve problems. In this next dimension of problems, it's about decisions and roles that people play when tackling a problem. Sometimes you start in one place and you take some steps and you eventually get to your target goal, right? And in hindsight, you discover that maybe you could have taken a shortcut, right? Some problems look like this. Other problems are a little bit less counterintuitive, more incoherent. So you might have one step that you take that actually ends up preventing you from reaching your final goal, but an earlier step you take actually helps you get to that goal, right? Sometimes the path you take towards a goal is not obvious. Other times it's complicated. Other times you have a decision to make. You take one path like this, or you can take another path that doesn't get you as far. But when it comes to your end goal, if you take this path that you think is correct, you get to this one outcome, but that might not be what you want. You take this other outcome, that other path that requires you to take a few steps back, you actually get to a better outcome. So in terms of solving problems, the path forward also is kind of unclear. And the last example I'll talk about is just sometimes you have to wear two different hats when you're solving a problem. And then each of these tasks takes on more work, and you just keep going, and you have to wear different hats. And you have to juggle all these responsibilities. So all this to say, problems can be solved in different ways, and the ways that you take to solve that problem require different resources, human resources, financial, technical resources, etc. And the last dimension, the third one, I'm going to show you an image, and I want you to think about what is the first thing that you see when you see this image. Is it one thing or the other? Is the first thing you saw a duck, or is the first thing you saw a rabbit? Sometimes when you deal with problems, there's different ways to look at that same problem. In this case, there's probably two ways to look at it, a duck or a rabbit. But sometimes the problem has features that might look like it is worth exploring in areas where it's more scarce, where there's less promise. So as someone who is a problem solver in the field of synthetic biology, there's these three dimensions that we deal with. The fact that we need to work with different kinds of knowledge, the fact that we need different resources to solve problems, and the fact that problems that are complex have different angles that they can be viewed with. So let's apply this to a synthetic biology example. Say we want to create a molecular diagnostic tool using a paper-based cell-free system. In this example, we're looking at a picture from Keith Party's work. And you might approach this problem with the experimental approach, where you want to design the cell-free mixture to have the right components to be able to respond to a viral RNA sample, for example. Maybe you want to use the paper-based diagnostic to detect metals, to detect antibiotics, hormones, etc. You could approach this from an experimental perspective. If you're mathematical in your training, you might think about this in the terms of how the reagents, the components of your cell-free mixture will behave such that you can predict how long it takes for a signal to appear, whether you need to modify the ribosome binding strength to increase the reaction rate, etc. You might think about this synthetic biology problem from a regulatory point of view, from a patent point of view. How do you actually pick the right molecular components so that you don't infringe on existing patents, for example? And how do you develop this such that it can be approved by the FDA, for example? And even further, perhaps you're more interested in how people on the floor working with infectious outbreaks, for example, would actually use a device like this. What kind of timing do they need? What kind of training do they need? How can you prepare the product to be able to be welcomed by the market right off the bat? So for this presentation, I explained problems first, and I explained it in the context of synbio because the problems are hard to deal with in terms of their complexity. And when we think about problems that can be solved partially or fully by synthetic biology, the point I want to make is that you require different forms of knowledge. There's the biochemistry, there's the mathematical modeling, there's the social sciences, the entrepreneurship. There are these different dimensions to the one problem that we could be trained on. And my question is, at CSBERG, how can we prepare students to know that there are these different dimensions for synthetic biology and allow them to better pick career paths that allow them to make the best use of their skills and strengths? So the first thing I'll talk about is what is CSBERG. And then I will talk about this design, build, test, learn cycle that we use in synthetic biology, but also in CSBERG from an educational point of view. So there's currently one cycle that we've almost finished completing for this DBTL for developing a course for synthetic biology. I'll walk through it. And then there's a second cycle that I'll briefly touch upon at the end so that you know where CSBERG is moving towards. And throughout the presentation, there's 3 questions I think that, if you get sleepy, you can ask these questions to yourself in the meantime. What do you think a synthetic biologist should know based off of your graduate experiences or industry experiences? What did you like about your favorite class? And what makes it hard for you to learn? Generic questions that you can think about throughout. So CSBERG is a nonprofit organization in Canada. And the goal that we have, our mission, is to search for how to best train synthetic biologists. It is not to be the best at training synthetic biologists, but to simply search for how students learn best for synthetic biology. And what we hope to do to carry out this mission is to create synthetic biology courses that are developed through data-driven iterative design cycles, integration of actual theory in the literature, and talking to folks like yourselves in academic labs, to startups, to industry, to learn more about what people want their practitioners or symbio to be able to do. The nonprofit is structured as follows. Blue, I know Amy is joining me in the call. Amy and I are the program directors who lead the executive team that lead the lead instructors who are the main teachers and the business team. And we're supported by our academic advisors across Canada, different universities, as well as our board of directors. And the team looks a little bit like this. It's a very diverse team with folks in Montreal, Ottawa, Toronto, Calgary, British Columbia. Like there's a lot of people across Canada. Many people come from iGEM, many don't. And there's also 10 more people on the top 3 rows that aren't here because they were recruited recently. All this to help us develop these courses that are highly interdisciplinary in nature. And to highlight that interdisciplinarity, here is a rough schematic of where our members lie in their responsibilities. In green, we have board of directors. Blue, we have our exec team. Purple, we have the lead instructors. Red is business team. There's more people here, but they're not displayed right now. Black are the instructors for the core program, which I'll talk about in a bit. And yellow is the high school team. So there's a lot of overlap in people's responsibilities. And so in CSBERG, if you are interested in joining, or if you just want to know, it's a highly interdisciplinary cross-functional team. So for the presentation, the first thing I'll talk about is the first part of the DBTL cycle for developing a synthetic biology course. And so we're going to deploy a mixed methods research approach where we combine quantitative numerical data with qualitative textual data. For mixed methods research, there's approximately 5 steps that can be proceeded through that we usually follow for all of our research. First is, of course, to formulate the research question. And for this particular step, the question is, what is the state of synthetic biology education from an iGEM lens? The limitation, obviously, is that not every student in Canada that does synthetic biology has gone through iGEM. However, it is a relatively representative portion of a large portion of the students for synthetic biology. So that's where we started. We had no idea what synthetic biology education was like in Canada. So this is how we started that question to explore it. With that question, we developed a series of survey questions, interview questions that we submitted to U of T's Research Ethics Board office to get approval to cover these kind of topics. We then contacted the iGEM teams shortly after the 2019 iGEM competition. So around 2019 in October, after that, we started contacting them. We then contacted them to do all the surveys, to do all the focus groups interviews. And we converted all those interviews into like written transcripts that we then coded using something called thematic analysis. So in qualitative research, thematic analysis is where you take those textual pieces of data and you systematically code them, which you can see here. You look at different paragraphs and you code parts of it according to themes. I'll show you some themes in a bit, for example. And then from there, you can do your more comprehensive data analysis and prepare the manuscripts. Before I keep going, so Ben, let me know if I'm taking too long. Just shout at me and I'll wrap up the presentation. Okay, so in the first study we did with these iGEM teams, we surveyed 17 of the 19 iGEM teams in Canada, and we interviewed them, 12 of them. And from these 12 transcripts, we were able to extract these 5 themes. Each theme has their own subthemes. The 5 themes are conflict and social cohesion, the dynamic interplay between the team's support network and the actual university. Third is developing skills that are complementary to academic training and lectures. 4, which is quite important, the lack of training for dry lab material, so computational mathematical stuff, and also human practices, so the social sciences and entrepreneurship, lack of training there. And 5th is the differentiation between how students learn synthetic biology working on a design team versus learning it in courses. And I will share these slides with you folks afterwards. You can look at these subthemes in more detail if you'd like. But what these subthemes kind of synthesize with this thing we call the FTSB, the Framework for Transdisciplinary Synthetic Biology Education. And to walk you through this, basically the FTSB is a framework to develop synthetic biology courses and programs that we propose, that we want to use in CSBERG that other people are free to use. And there's 4 elements to the FTSB. The first is the knowledge domains. You need to outline what exactly you're teaching students. We're familiar with the wet lab content, probably familiar with the dry lab content. These areas have more structured curriculum that already exists. What is less structured are RRI and business. So RRI stands for Responsible Research and Innovation, and then there's business. So this entire domain of synthetic biology is just very poorly taught in Canada, but it's such an important aspect for synthetic biology in terms of translational research. So we have a dedicated element to specifically say, if you're going to train someone to be an interdisciplinary practitioner, they should probably know some basics in this yellow domain. Now, what connects these knowledge domains together at the core, at the center, at the beginning of a course or a program is disciplinary humility. The idea that your one discipline, biochemistry, for example, is not enough to solve issues that impact people, that impact the environment. You need to incorporate other fields of knowledge. And getting students understanding that right off the bat is critical, because what we've heard in talking to other professors is that by the time students get to 3rd or 4th year, some of them already have this belief that like you can use biochemistry or like experimental synthetic biology to address most of the world's problems, when in reality, there's so much more to it than just this one siloed discipline. So we start with disciplinary humility there, and we branch out. And this IDSF stands for the Interdisciplinary Science Framework. It's a published work piece that we use to guide students' ability to collaborate. 3 is learning progressions, where knowledge gets more advanced over time in a coherent manner. And 4 is this backdrop where students are learning project-based courses, basically. So based off of the FTSB, we then thought, okay, the 3 knowledge domains, what exactly are we teaching students? So we had to come up with learning outcomes for a student that would be learning in each of these domains to develop a course. There's a lot of details on the screen, but to summarize, the experimental learning outcomes have to do with the ability to prepare cloning workflows and analytical techniques to actually monitor the outputs of these plasmids. For computational learning outcomes, it is learning how to model deterministically, mostly, with some stochastic commentary, and also learning how to, the basics of the bioreactors, the stoichiometry, ODEs, and a little bit about machine learning for how to process large data sets, perhaps related to bioreactors, but also proteins. And finally, for implementation sciences, which is that third domain I was talking about, that yellow area, essentially it's helping STEM people think at a more systems level, in a more translational level. So the actual course looks like this. There's two courses we taught. For the first course in B1, there's two streams of students being taught at the same time. The first stream is week 1, 2, and 3, where we teach them like through lectures, how to use benchling, how genetic circuits work, and how to like basic metabolic engineering principles. And for the computational stream, bioreactors, ODE modeling, and basic machine learning. And at the end of the course, week 4, we provide them a small design challenge for students from both streams to come together to work on a design challenge that will kind of test their skills in each area and their abilities to collaborate. For SYNB2, again, that's technical topics, focus more on the social sciences, the science of communication, IP, and general business principles, culminating in a case study. So the first time we taught this, students had a hard time doing week 4's design challenge because we were putting them in groups with people that they'd never worked with before. So the second time we taught each of these courses, what we focused on was building relational resources, which is pretty straightforward in the sense that if you've worked with people before and you work with them on a new project, you're going to perform better than people who are just complete strangers. That's what relational resources are. And the way we built that was in iteration 2, for SYNB1, we had students form their groups much earlier in week 1. And after every class, we gave them time to connect with each other and share what they learned in each of the courses. For SYNB2, similar thing again, we formed the groups a little bit earlier. And every time they work on activities in week 1, 2, and 3, they'd be working together in their groups that they would be in week 4. So the research that we did for this course was divided as so. So SYNB1 and SYNB2 are these two different courses where all students will have some asynchronous videos to watch beforehand. They go through week 1, 2, and 3 where they learn content, and week 4 is when they're challenged. Over time, these stars represent where we survey students. So we ask them to fill out a survey, like a questionnaire, to rank how confident they feel about a certain topic. So these are attitudinal scores, we call them. So the yellow stars are the questionnaires, and the orange star is the focus group that we do at the end with the design team that work on design challenges together. So we're in the midst of like analyzing this data as we speak, and also I wanted to share some of the data with you, the quantitative data. In this case, for example, right now we're looking at some of the data for wet lab. The left panel is corresponding to this one measure, where students rate their confidence in their ability to design plasmid constructs. B is the ability to propose cloning workflows, and C is their ability to actually think of ways to measure the outputs of the genetic circuits. We see that at the beginning of the course in survey 1, their scores are low, they're like around 4, and over time they increase to about 8 for all 3 of them. For the dry lab, again, we see patterns where students' scores are increasing over time for these 6 different learning outcome metrics. It doesn't really matter what the outcome is, but what matters right now is that it increases over time. And I'll explain why it doesn't matter exactly what they are. For the SYNB2 IS stream, in this question, we ask students, how knowledgeable are you of the roles of these folks in the innovation pipeline? And we see, of course, the students are mostly science students and engineering students, so they're going to have high scores right at the beginning, and it stays high. But for these other roles, where they're not really taught about how regulatory bodies work in their courses or how IP lawyers work in their courses, of course, it starts low and increases over time. The reason why I say it doesn't matter in particular, like what is increasing over time, is because of something called the Hawthorne effect, which is this idea that there's going to be changes in people's behavior, so how they learn and how they report what they're learning, because they're under observation, which is going to bias the outcomes of the research study. So all this data, I would not assign like statistical significance to them. The best they can do is act as a compass for us when we do the qualitative research, where there's more rich data to mine through. So this data is helpful for us in understanding whether students do feel confident or not. But we can't say that like for like this one for B, right, for implementing machine learning techniques, that their ability to do it increases over 50%. We can't say stuff like that. All we know is that they feel like their ability to do it increases over time. This is where qualitative data is really helpful. And I just wanted to quickly show you what that qualitative analysis looks like. Earlier when I was talking about the surveys we did with the iGEM teams, this was done by hand. So we printed out all the transcripts on paper and used highlighters to like code everything. What we've been able to do instead is this program called Deduce, which I will log into. And this is a great software. I don't know if you ever will do qualitative research, but I love this software. And the idea is that you upload all of your text transcripts into your media. And what you also do on the left-hand side, you see here, is upload all of your codes that you will code information in these transcripts with. So here are some of the codes that we have already for our themes in the green, and the blue, our sub-themes. And what we've done is exhaustively, we've gone through all 21 of these transcripts, and we've coded every time a student will refer to one of these codes. And what the software will do is keep track of all the excerpts and what codes are applied to them, which you can then export to an Excel sheet to do more of a quantitative analysis. So in this Excel sheet we see here from the excerpts, there's about 450 excerpts with this many codes applied to different areas. So we see on this axis here, these are the different codes that we have. On this axis here, these are the 21 transcripts. And in this sheet in particular, it's how many times that code has been applied in that transcript. So if we're interested in understanding what are the learning barriers for students, then we can ask the question, well, what's the most frequently populated subtheme? In this case, for example, time constraints is a big issue, as well as students not having enough time to be able to digest the content, probably because they have time constraint issues. And you can see how by combining this qualitative quantitative part, you can kind of have more intuition about what needs to be improved for learning barriers, what worked well, how can we have better interdisciplinarity, and what exactly in the course do we need to adjust. Another thing you can analyze is which codes will coexist with which other codes. And this will help you understand which codes are basically really similar, that you can just combine them together. So going back to presentation, basically done. The last thing I wanted to share is this cycle number 2. What are we doing to develop the course again? Because we've now taught the SYNB1 and SYNB2 courses each twice, so 4 in total. Now our question is, how do you develop a course even better than those two? And so for this, I'll walk you through the new syllabus we've developed for a course called SYNB3, Introduction to Integrative Synthetic Biology. So SYNB1 and 2 were 2 separate courses, where one was teaching wet lab and dry lab, and the other one was teaching these social science entrepreneurship concepts. In SYNB3, we've put them all together to create this course where you have 3 streams of students, so 40 each, are learning content across 5 weeks this time, and they work on a design challenge for 3 weeks instead of 1 week. So it's a much more comprehensive course where students can have full collaborative freedom. And in case you're curious, the content we're teaching this time, I'll zoom in a little bit. So again, these are being taught at the same time. Week 1, wet lab learns essentially like how to use benchling. And that's where they'll learn their basic molecular cloning through our benchling activities. To then proceed on to work with circuit design, pathway design for medical engineering, genome assembly for metabolic engineering, for example, and they end off with a case study. For computational, they first learn scientific computing, so we'll get them comfortable with Python. They then proceed to learn about basic concepts for bioreactors. In week 3, we combine these areas where we teach them, okay, if you have a lot of data, how do you properly work with it in the Python pipeline? Expanding that, what if you have protein data sets? How do you work with those, again, using Python, leading up to a case study? And lastly, in information sciences, we first start off with what exactly is information sciences, because these are STEM students. Introduce them to alternative research methods besides our scientific method we use for quantitative mostly, and then bridge their knowledge with issues in biosecurity, in how to actually implement innovation, pitching, IP, etc., all culminating in the design challenge. And the design challenge we have this time, so we're working with Kevin Chen from Hyacinth, where essentially students are working on a metabolic engineering project where they have to create rare cannabinoids. And in this design challenge, I won't go into detail, but essentially it lays out different types of design challenges, all the same topic, but different flavors. There's automation flavor, manufacturing, and medical. So students will have a choice this time. Instead of having only one, they get a choice to pick between 3 different design challenges to exercise all of the content they teach in the actual course. So I've been talking a lot. I'm hoping to have discussion just like on some of the stuff I've talked about. To sum it up, what is CSBERG looking to do in the future? What we're looking to do is to continue developing and building on this FTSB, this framework for teaching synthetic biology, by establishing our core programming. So SYNB2 is our main course, our flagship course. And then once that is solid, we then develop the advanced courses. So some other core courses we might develop are automated experimentation, like a bigger intro to scientific programming. And for our advanced courses, some of the first themes our instructors want to teach are cell-free medical diagnostics, bioreactor data analysis, and protein-based metal recovery, which is my content that I work on for my PhD. And a quick shout out for people who are watching this in the recording for undergraduate students. SYNB2's course enrollment drops tonight, 9 p.m. EST. Here's a link. And I'll stop there. That is the presentation. We're at 12:42 on time.
Yep, you were just in time.
Excellent.
So thank you, Patrick, for your very nice presentation. From now on, the questions and discussion session is open, so feel free to ask your questions and comments to Patrick. And we will ask you to use the raise your hand function for it to be more easier to modulate, and write your questions directly in the chat box. Yes, Ahmad, you can go.
Thank you so much, Patrick, for this very nice presentation. First of all, I would like to thank you for having accepted this invitation and kind of present to the Synthetic Biology Club organized by our lab. My name is Ahmed Saleh. I'm a professor, a new professor in Malaya University, and principal investigator of this lab of Abdel-Mawgoud Synthetic Biology Lab. And I'm also teaching a synthetic biology course for undergraduates. So that's why your presentation is really or extremely interesting to us, since I'm developing the course for the undergraduate program of biochemistry. Certainly I would be able to implement many of your nice ideas. So, but I have some questions that came to my mind when you were talking about your very nice iterations of developing synthetic biology 1 and 2 and now 3. So what's your imagination of the synthetic biology course? Where it is placed? Is it a program, a separate program, or it is a culminating course at the end of biochemistry program, for example, or other programs? So because this will define the intensity and the density of the content of the course, certainly we know that synthetic biology is not a course. It is an interdisciplinary science that cannot be summarized as a separate entity. It's something that's branching in everything. And if we want to teach synthetic biology, we cannot do that, in my opinion, as a separate course or a separate program. We instead need to deliver a closure summaries or concepts, to deliver some concepts to the undergraduate students about what is it about synthetic biology, how can we apply. This is so I want to hear from you, how do you place synthetic biology? Is it a separate program or it is a course within other programs?
That's a good question. I'm going to share my screen again. Reviewer number 2 had the kind of the same question, like how do you actually implement some of these suggestions? Because reviewer number 2's critique was that, you know, you're reimagining a university setting to be able, if you want to implement this, you have to create like a new cross-disciplinary, interdisciplinary faculty to be able to actually implement this. It's very difficult in a university setting, right? So my answer to your question is there's 2 parts. One part is you could implement some of these ideas in this paper in just a series of courses, perhaps as a minor or as a specialization. And you can do what Western University does and just have a series of biochemistry courses with add-ons that will make it so that you're teaching students more of these synthetic biology concepts. So you would have like a second-year course that introduces concepts in synthetic biology, and then you can have perhaps third-year courses, fourth-year courses where you get into more specific topics. So we like to use genetic circuits. You could teach courses on just genetic circuits and how to build them and how to design them. You could develop a course for how to model them mathematically with ODEs and predict their behavior. The first part of my answer to your question is you could keep the existing courses, modify them to have synthetic biology elements, perhaps referring to literature, and add new courses that will build off of like second-year concepts. That's the first idea. But I think that this idea is the non-ideal case because what we see at Western, for example, is that it's still very experimental heavy, right? If I'm someone who has like mathematical training, like pure math, for example, I can't go into that program and be able to retain my math background, right? So what I personally envision, you know, in like a world where there's unlimited funding for this, is like a university setting where you enter this program and right off the bat, the faculty of quote unquote synthetic biology already has first year and second year courses that are co-taught by folks from other disciplines like engineering, sciences, and social sciences. And you have dedicated core teaching staff that are responsible for integrating knowledge. And as students progress through this kind of undergraduate program, that core teaching staff is responsible to make sure that their learning progressions, say they want to teach students to know how to do genetic circuits, like that's the core skill you need. The core teaching staff is responsible for making sure that through year 1 to 4, they keep building on this concept. And they draw on the expertise of specialty professors to, you know, provide the latest insights from a research perspective, industry, etc. There needs to be a core teaching staff that's responsible for students the entire time. I don't think it can be like a bunch of professors coming together and like a non-teaching staff like coordinating people. It doesn't, you're going to lose out on that interdisciplinary.
Mm-hmm. Nice. So we have such scenario in our program, which is called concentration, which is concentration or tracks, where we can, for example, add a track of synthetic biology or synthetic biologists, where in this track, some other courses are prerequisite for some. For example, some, as you said, for chemical, we can add some chemical engineering courses, some mathematical modeling, some some human sciences, sorry, some human sciences and so on, so that he can graduate as a synthetic biologist at the end. Yeah, nice, very nice. Thank you.
Hi, Patrick. I had a question. It's more to help you sell. For people that don't know, when they are given the 3 courses of SYNB1, SYNB2, and SYNB3.
When is it? SYNB1 and 2 are done. So those were like research projects we wanted to iterate to learn. SYNB3, the enrollment opens today, 9 p.m. EST. It's primarily designed for undergraduate students. So most of our enrollment for SYNB1 and 2, and this time as well, will be 2nd and 3rd year students, some 4th year. We don't have graduate level courses yet. Those will be probably the advanced programming. But yeah, it's today, 9 p.m. EST, and it'll last until April 18th.
To add on very quickly too, SYNB3 runs from May to June on the weekends, just because we're all students. So obviously we're a little busy during the week, but it runs on the weekend, Saturday and Sunday, for about 2 hours a day. And I think it's the full 8 weeks that the course will last.
So how many hours in total for the course?
It's about 2 to 3 hours a week, in total 24 hours.
Very nice. Thank you. Yeah. Very interesting. And how do you conduct, so there are wet lab and dry lab and some other courses. So how do you ask students to do the wet lab, for example?
The wet lab, so we don't have like in-person physical training for like how to, mini prep. It's all focused on how do you design experiments properly, and how do you prepare ahead of time for troubleshooting issues. I think, I don't know, from my experiences and a lot of the instructors' experiences, I think half the time we're troubleshooting, trying to figure out what's going on, instead of just doing the experiments. So that's the focus for these students. I think in the future, we're going to expand to get more funding, and then become a physical location where we can actually teach physical, experimental biology. That's a future thing, though.
Yeah, nice.
Yeah, very interesting, because as an iGEMer of the team of Laval University, this is something I could share them, because we have a lot of new people that joined the team. So having new information and formation on synthetic biology could be very useful for them.
I'll also comment. Laval's iGEM team is solid. You folks have performed well for the past couple of years. A lot of the folks on CSBERG come from iGEM. So if you find that your students need extra support, perhaps you want to hear past judges comment on the project, we can do that. We have several judges on the team, or past judges at least. We can help with project decisions, how to present the project, all sorts of things. So just let us know if you do want that kind of extra support, because we can help out.
Yeah, very nice. I'll take note.
Yeah, I've judged 5 years now.
Yeah, I've recognized that Patrick was also a judge.
Yeah.
Technically, we've reached the 10 minutes of questions and discussion. So if there is no more questions, I think this concludes the activity. And so, yeah, Ahmad. Your microphone is off.
I have just one last question. So what's your message, Patrick, to professors who are across Canada teaching synthetic biology courses? So what's your message to them in order to concert our efforts and develop something that's more solid and that's more satisfactory to the expectations of students who are taking the courses of synthetic biology?
I think some of the best teaching methods that students enjoy from our experiences are these projects where they have the ability to kind of explore what they're interested in. So if you want to maximize the student's ability to learn something in your course, of course, teach them the fundamentals, the core concepts, and give them the opportunity to run free with that knowledge, and try using benchling to design a cloning workflow, go read the papers and propose a new type of project, and kind of encourage them to use their imagination. I think that's quite important, because I think through grad school, at least for myself and some of our instructors, our favorite experiences in grad school is the ability to take control of our projects and take it in a direction that we think makes the most sense, that the data points at. So if undergraduate students have the opportunity to fail, but learn to keep moving forward, I think they'll be the best use of their time, because the real world is filled with failures, I think. So we try to do in our design challenge. They will probably fail different parts of the design challenge, but we tell them why, we explain to them, and then in the future they can try again in labs.
Thank you so much.
Yep.
That's it.
So thank you everyone who joined and participated in the activity, and most importantly, thank you, Patrick, for your time and presentation.
No problem.
And as a quick reminder, if you want to present your work or an article, you can always register as a presenter on our website, and otherwise you can register as an attendee. Also, every last Monday of each month, we have a new presentation, and the next one will be on April 25th, and we'll announce the next presentation and presenter the second week of that month on the website. So goodbye, everyone, and have a nice day, and see you next time.
Thanks, everyone.